Assalamu alaikum and welcome everyone to the ninth lecture of Molecular Biology ASCP preparation series. The molecular science part is divided into four sections: nucleic acid chemistry, basic molecular theory, biochemical reagents, and genetics, which together make up 20 to 25% of the total molecular biology ASCP exam. In our previous lectures, we covered all MCQs related to the nucleic acid chemistry. We then moved on to the basic molecular theory section and completed the replication parts, transcription parts, exon, intron, and splicing part. Now we'll delve into translation section. For those who missed the previous lecture, don't worry. I have added the links to those lectures in the bio below, so please check it. All right, let's move on. Okay, now let's focus on the question related to the translation in eukaryotic cells. A review ASCP question from previous exams. Okay, so the first question we have today is which RNA sequence signals the start of translation in eukaryotes? Let's move on. Okay, the RNA sequence that signals the start of translation in eukaryotes is the AUG codon. Think of AUG codon as the start button that tells the ribosome where to begin building the protein. Okay, let's move on. Okay, the next question. Which eukaryotic ribosome subunit binds to the messenger RNA first during translation initiation? All right, the correct answer is 40S. The small 40S ribosome subunit scans the messenger RNA for the start codon AUG. So once it identifies the start site, then the larger 60S subunit joins to form the complete 80S ribosome. All right. The 40S ribosome subunits bind to the messenger RNA first during translation initiation in eukaryotic cell. So we need to think of the 40S subunit as the Scot that locate and attach to the messenger RNA, setting the stage for the larger 60S subunit to join and start building the protein. Okay, let's move on. Okay, the next question is what is the primary site of translation in eukaryotic cells? Is it nucleus or cytoplasm? For sure, the primary site of translation in eukaryotic cells is the cytoplasm. This is where the ribosomes are located, and it is the main area where proteins are synthesized from the messenger RNA templates. Okay, let's move on. All right. What is the function of eukaryotic initiation factor 4E? Okay, the function of eukaryotic initiation factor 4E is to bind to the 5' cap of messenger RNA. Think of these factors as the key that recognize and attach to the special cap at the beginning of messenger RNA molecule. So this step is crucial for initiating translation as it helps the ribosome find and attach to messenger RNA to start building the protein. Okay, let's move on. Okay, the next question is which molecule delivers amino acids to the ribosome during translation process? This is like a gift question when it comes in the exam, right? For sure, transfer RNA is the molecule that delivers amino acids to ribosome during translation process. We need to think of transfer RNA as the delivery truck that carries specific amino acids to ribosome where they are added to the growing protein chain. Okay, let's move on. All right. The question here, what is the function of eukaryotic elongation factor 1A? Actually, eukaryotic elongation factor 1A works like a delivery truck. It brings the correct transfer RNA, which carries amino acid to the ribosome. This makes sure the right amino acid is added to the growing protein chain at the right time. So the correct answer here is delivers aminoacyl transfer RNA to the ribosome. All right, let's move on. So the next question is, which eukaryotic translation termination factor recognizes stop codons? All right, the eukaryotic translation termination factor that recognizes stop codon is ERF1 or eukaryotic release factor 1. Think of eukaryotic release factor 1 as the checker that identifies the stop codon on messenger RNA, signaling the ribosome to stop adding amino acid and release the completed protein. All right, let's move on. In eukaryotic translation, what is the rule of poly A tail? All right, let's see the correct answer. The poly A tail at the three ends of messenger RNA stabilizes the transcript and enhances translation by what? By interacting with the protein that help ribosome binds. All right. In eukaryotic translation, the poly A tail acts as the protective cap, preventing degradation and ensuring efficient protein synthesis. All right. So the correct answer here is stabilizes messenger RNA and enhance translation efficiency. Let's move on. Okay, next question is, what happens at the ribosome P site during translation? Okay, let's move on. During translation, the ribosome P sites hold the growing polypeptide chain. Think of the P site as the workspace where the chain of amino acid is built up step by step and each new amino acid is added. All right, let's move on. The next question is, what is the role of microRNAs in eukaryotic gene regulation? Actually, microRNAs play a crucial role in eukaryotic gene regulation by what? By repressing translation or degrading target messenger RNAs. We need to think of microRNAs as the molecular brakes that can slow down or stop the production of specific proteins by either preventing the messenger RNA from being translated into protein or by causing the messenger RNA to be broken down. Okay, let's continue. Which eukaryotic elongation factor facilitates ribosome translocation? The correct answer is C. Actually, eukaryotic elongation factor 2 is responsible for translocating the ribosome along the messenger RNA, shifting the transfer RNAs from the A and B sites to the P and E site respectively. Okay, let's move on. The question here is which enzyme catalyzes the formation of peptide bonds during eukaryotic translation? Alright, the correct answer is peptidyl transferase. This peptidyl transferase found in the large ribosomal subunits 16S. It acts like a builder. It links amino acid together to form a protein chain, ensuring the growing protein is assembled correctly by catalyzing the formation of peptide bonds between amino acids. Okay, let's move on. Alright, the next question is what is the rule of Kozak sequence in eukaryotic translation? Alright, the consensus Kozak sequence represented as the 5' GCC GCC as we see in the picture, followed by the start codon AUG in eukaryotic messenger RNAs. This sequence help ribosome find the right starting point for translation. It is like a special signal on the messenger RNA that guides the ribosome to begin building the protein accurately and efficiently. So the role of Kozak sequence in eukaryotic translation is facilitate efficient start codon recognition. All right, let's move on. What is the energy source used during translation elongation in eukaryotic cells? All right, the correct answer is GTP or guanosine triphosphate. Think of GTP as the fuel that powers the process. It provides the energy needed for the ribosome to add each new amino acid to growing protein chain, ensuring everything runs smoothly. All right, let's move on. Please don't forget to subscribe to our channel to receive all the latest lecture. Okay, from here, subscribe, guys. Okay, let's continue. Okay, the next question is what is the role of eukaryotic translation initiation factor 2? Actually, the role of eukaryotic translation initiation factor 2 is to deliver the initiate and transfer RNA to the 40S ribosomal subunits. Think of eukaryotic translation initiation factor 2 as the delivery truck that brings the first transfer RNA, which carries the starting amino acid methionine to the small ribosomal subunits. All right? So this step is crucial for starting the translation process correctly. Let's move on. All right, the next question is what happens when a ribosome encounters a stop codon during translation? Actually, when the ribosome reach stop codon such as UAA, UAG, or UGA, special proteins called release factors bind to the ribosome. These release factors help release the finished protein and break down the ribosome. All right. So the correct answer here is release factor bind to the ribosome and terminate translation. Let's move on. Let's see the next question. How the ribosome messenger RNA interaction is stabilized during eukaryotic translation initiation? Actually, during eukaryotic translation initiation, the ribosome messenger RNA interaction is stabilized by interaction between the 5' cap and the poly A tail. All right. The 5' cap binding proteins and the poly A tail binding proteins interact to form a loop, bringing the two ends of messenger RNA close together. This circularization helps ribosome stay attached to the messenger RNA, ensuring efficient and accurate translation process. Okay, let's move on. Okay, the next question is, what ensures the accuracy of amino acid incorporation during eukaryotic translation? Actually, to ensure accurate amino acid incorporation during eukaryotic translation, two key factors are crucial. The first one is codon-anticodon pairing. The second one, amino-acyl transfer RNA synthetase specificity. Let's explain each one first. Okay, for the first one, codon-anticodon pairing, each transfer RNA molecule has anticodon that matches the specific codon on messenger RNA, ensuring the correct amino acid is added to the growing protein chain. For the second point, amino-acyl transfer RNA synthetase specificity, these enzymes act like quality control inspectors, attaching the correct amino acid to its corresponding transfer RNA. So, together, this mechanism ensure that the protein is built correctly. All right, let's move on. Okay, the next question is, which site on the ribosome does the amino-acyl transfer RNA initially bind during elongation? Alright, during elongation, the amino-acyl transfer RNA initially bind to the A site or amino-acyl site on the ribosome. Alright, let's move on. Okay, the next question is, which elongation factor prevents the premature hydrolysis of GTP during elongation? Okay, the correct answer is eukaryotic elongation factor 1A. Alright, let's think of eukaryotic elongation factor 1 as the carefully manager that ensure GTP, I mean the energy source, is used at the right time. Alright, it helps deliver amino acid to the ribosome and make sure the GTP is only used when in absolutely necessary, preventing any waste or premature use of energy. Alright, let's move on. What ensures ribosome diassembly after translation termination in eukaryotes? Okay, after translation in eukaryotic cells, ribosome recycling factors act like cleanup crew that disassemble the ribosome into its parts, right? On the other hand, GTP hydrolysis provide the necessary energy, just like fuel for the cleanup crew to do their job. Okay, so the correct answer here is ribosome recycling factors and GTP hydrolysis. Let's move on. Alright, the next question is, which component recognize the codec sequence during eukaryotic translation initiation? Okay, the correct answer is a small ribosomal subunit. This small ribosomal subunit is the component that recognizes the codec sequence during eukaryotic translation initiation. The codec sequence actually is the specific sequence for nucleotide around the start codon in messenger RNA, as I said earlier, and the small ribosomal subunits helps to identify and bind to this sequence to start the translation process. All right, let's move on. Okay, the next question is, what happens when the elongation ribosome encounters a defective messenger RNA lacking a stop codon. Okay, when the elongation ribosome encounters a defective messenger RNA lacking a stop codon, the non-stop decay pathway degrades the messenger RNA. Think of this pathway as the quality control mechanism that identifies and breaks down faulty messenger RNA, preventing the production of incomplete or non-functional proteins. All right, let's move on. Okay, the next question is, which of the following is the common post-translation modification of newly sensitized protein? Which one of them? The correct answer is phosphorylation. Phosphorylation is the common post-translation modification of newly sensitized protein. Think of phosphorylation as adding small chemical tags, phosphate group, to a protein, which can change the protein function, activity, or location within the cell. So, the phosphorylation, along with the other modification like glycosylation and acetylation, occurs post-translationally to regulate protein function and activity. All right? Okay, the question here. What is the role of eukaryotic initiation factor 4G in translation process? All right. The role of eukaryotic initiation factor 4G in translation is to act as the scaffold to bring messenger RNA, ribosome, and initiation factors together, as we see in the image. Think of eukaryotic initiation factor 4G as the organizer that help assemble all necessary components at the right place and time to start the translation process efficiently. All right? Let's move on. Okay, the question say, which molecule ensures proper folding of nascent polypeptides during translation? The correct answer is chaperone proteins. Chaperone proteins are like the cell quality control team. Actually, during translation, when new proteins are being made, chaperone proteins help them fold correctly. Okay? Think of it like making sure a piece of string is neatly folded instead of tangled. So, without chaperone proteins, the new proteins may not fold it properly, which can cause problem for the cell. All right? So let's move on. Okay, next question is, what role does the ribosome 16S subunit play in translation? Actually, the 60S ribosome subunits play a crucial role in translation by catalyzing peptide bond formation. This means it helps form the bond between the amino acid to create a growing protein chain. This is the last question we have it today. Thank you. Thank you for your attention. I hope you enjoy the lectures. Please don't forget to like, comment if you have any question, and subscribe to our channel to receive all the latest lecture. Salam, and see you in the next lecture.